In this session, we're going to talk about skeletal muscle contraction and some of the microscopic parts of the muscle. And so basically we're talking about how a muscle, or the myofilaments, the actin and myosin, move within our muscle fiber. So here you can see in this particular diagram, we've pulled out a myofibril from our muscle fiber, and you can see some of the things that we're going to label here. The thicker purple strands are the thick or myosin filaments. You can see we're there, and there's also the blue line depicting the thin or actin myofilaments, as you can see throughout this myofibril. And we're going to talk about how they're arranged here within a single myofibril. Another definition, or probably a good place to start with as we're sort of defining the myofibril and the parts of the myofibril, would be the sarcomere. And you can see that labeled right along these two areas right here, sarcomere. And they go from what we call from a Z line to a Z line. So the ends of the sarcomere kind of make a zigzag pattern, as you can see right here and right here. And this zigzag pattern constitutes what we call the Z line. Within the middle of our sarcomere, we have where we just have the myosin and those kind of globules of proteins constitute what we call our M line. And that's in the very center of the sarcomere. The M line sort of anchors, if you will, the myosin filaments. And as a contraction takes place, it's those myofilaments that kind of stay stationary, and our actin filaments are actually going to contract and shorten up, kind of like almost an accordion effect. Within that, if we're to look at a myofibril under a microscope, we'll have areas where more light passes through the microscope and areas where less light passes through the microscope. So our first we have is what we call our eye band. Okay, and our eye band is just an area within the myofibril where there's just the thin filaments, or the actin filaments. So a little more light will actually pass through. Then we have, I'm going to skip to the H zone. And the H zone is the area where we just have the thick filaments. So right here in the middle is where that H zone would be, and our eye band is over here. So that would be a little bit thicker particular area, and less light would come through because myosin is a thicker protein than the actin. Next we have what we call our A band, and so that's a pretty broad band, as you'll be able to see, and you could, as you might guess, we're going to have an overlap, as you can see, where the myosin protein right here gets overlapped with the actin protein, and so even less light is able to come through our myofibril in that particular A band. And so here you can see just a pictorial graph down here highlighting those things. On the next slide here, I have both a microscopic view of actual muscle tissue that's been stained, and then the pictorial drawing right below it. So as you can see, here are the Z lines labeled for us. Not labeled, the M line typically runs right down the middle. And then our bands and zones that we talked about regarding if more light is able to pass through or less light passes through, based on our I bands, our A band, and then right in this area is where our H zone would be, where there's just myosin. So that's a little bit about the arrangement of the myofibril. Keep in mind, and we'll talk about this in more detail later, um, as we zoom in, that there's going to be even more interaction. So just taking a look closely at the myosin filaments. Remember, the myosin are the thicker ones. Um, the thicker protein, the myosin, you can see is right here in the middle. That's more of this darker purple. And you can see part of the protein of the myosin comes off and forms what we call a cross bridge head. These cross bridge heads are going to actually at times come up, make contact with the actin, and actually act like a ratchet, if you will, pulling the actin along. The actin itself, obviously thin filament, and you can see the actin. There's four of them, four different actin molecules here. The actin actually itself is these little beads, or they look maybe like a string of pearls that kind of twist around each other. And then we have two other proteins that are involved with the, or interact with the actin itself. And those proteins are the troponin and the tropomyosin complexes. So the troponin is going to be the head of the end of the complex. You can see one there, you can see one there, you can see one here. So we have that troponin complex that's going to be involved with some interactions that come place in the future here. And then the second thing is the tropomyosin, and that's these long bands that actually connect to heads. And so just like the actin kind of twists around each other, this troponin and tropomyosin complex twists around the actin itself. And if you look real closely, you will be able to see in future slides really closely at each actin molecule, there's a little kind of almost purplish marking on the actin, and that's going to be what we call an active site. And we'll talk about that right now or a little bit later. Right now you can't see it as the troponin tropomyosin complex is covering it up. So to start with our uh, contraction of a muscle, we need to know a few more things about how a nerve cell interacts with our muscle. So the neuromuscular junction is just that. A junction is the place where two things meet, kind of like a road, like Highway M and Highway 16. There's a junction where they meet. Well, neuromuscular junction is where a motor neuron, and in this case a muscle, come together. Another name for a neuromuscular junction would be a synapse. Is the same type of term for the neuromuscular junction. So here you can see, we have four neuromuscular junctions or synapses, or you can see it here kind of blown up as we have kind of zoomed in. So this is a site where the axon, the motor axon, the terminal end specifically, meets up with the muscle. Okay, so some other specific parts, we got our motor neuron. You can see that coming down here, and we got different terminals, which I'm erasing right now. And so we'll talk about, is it better to have one of those, two of those, four of those, 12 of those? We'll talk about each one of those motor end plates and those in those junctions. The motor end plate itself, if you take a look where it's zoomed in, the motor end plate is actually found on the muscle cell. And note, as you can see in this particular synapse or this neuromuscular junction, the neuron does not even touch the muscle itself. There's a little space in between, and that space is called the synaptic cleft. Okay, and so we need that space so we can have a diffusion of some neurotransmitter, and we're going to talk about acetylcholine specifically. But we have synaptic vesicles, and we have those are produced in the neuron. So you can see those synaptic vesicles up here. So the Golgi apparatus and the endoplasmic reticulum make some sort of neurotransmitter, some sort of chemical, and that chemical gets stored in a vesicle. We learned about that earlier this year. So we have a synaptic vesicle, or vesicles that are in the synaptic end of our neuron. So if you take a look, all those vesicles are going to be located in the very tip here. And inside those synaptic vesicle is some sort of neurotransmitter, some sort of chemical that is going to carry the message from the neuron to the muscle to get the muscle to contract. And the one that we're going to focus on primarily is called acetylcholine. Sometimes we just abbreviate that and call it ACH. And that's one of the chemicals that is directly involved with muscle contraction. So we'll end there with this particular session, and we'll talk about how this muscle contraction actually takes place in the next session.